Hi students, welcome to the sessions of oral pathology. I'm Dr. Gopi Krishnan discussing oral pathology with you. Okay, we are here with a new topic that is our dentinogenesis imperfecta. So we are discussing structural anomalies of the tooth. We have already seen amelogenesis imperfecta in the previous video, and this is about dentin. This is about dentinogenesis imperfecta. Okay, so dentinogenesis imperfecta is the developmental anomaly that is affecting your dentin. Okay, here the dentin is affected. So in the previous video we have seen how the enamel is affected, what are the clinical features and all. Okay, oh, see this tooth. Okay, see this, you can see this picture. I think it is completely brownish. Okay, it is brownish in color because of the defect in your dentin, not in your enamel actually. Okay, so it's brownish tooth we call hereditary opalescent tooth. Okay, opalescent in color, opalescent hue. Okay, so let's see dentin on dentinogenesis imperfecta. So what happens in dentinogenesis imperfecta? So the dentinogenesis imperfecta is the developmental anomaly of your dentin where the defect lies in the protein. Main protein which is affected is your collagenous proteins. The collagenous proteins or the collagen is affected. The gene which is mutated in this dentinogenesis imperfecta, the collagenous genes which is mutated is called 1A1 and called 1A2. Okay, so collagen 1A1 and collagen 1A2, that is called for collagen. Okay, these two genes are predominantly mutated. Okay, so that will cause a defect in the production of collagen. Okay, and you know that dentin contains collagen. Enamel doesn't contain collagen. Okay, enamel contain only amelogenin and other enamel proteins. Okay, but dentin contain collagen. Okay, dentin, bone, and all the other hard structures contain collagen. Hard and soft structures contain collagen, but enamel doesn't contain collagen. So dentinogenesis imperfecta is mostly due to the genetic defect that is affecting your collagen gene, that is COL1A1 and COL1A2. Okay, and the other known collagenous genes, sometimes other known collagenous genes proteins or genes will be affected. That the main example is dentine sialophosphoprotein. Okay, DSPP, that is dentin sialophosphoprotein. There are other also dentin sialoprotein, dentin phosphoprotein, dentin glycoprotein, many are there. Okay, dentin sialoprotein is there, dentin phosphoprotein is there, dentin glycoprotein is there, and then in collectively we said dentin sialophosphoprotein, DSPP. Okay, so these two genes are the major responsible factors for amelogenesis imperfecta. Okay, now you know that the gene COL1A1 and COL1A2, they are also responsible for one another condition, another developmental anomaly, that is osteogenesis imperfecta. Osteogenesis imperfecta, this we will see when we are discussing bone. Okay, so osteogenesis imperfecta is the developmental anomaly of your bone. Okay, it's a developmental anomaly of the bone, and the same gene is involved in the dentinogenesis imperfecta. So in the previous classification, in the old classification, I say old classification was given by Shield. Shield has given the old classification. According to him, there is three types of dentinogenesis imperfecta, that is type 1, type 2, and type 3. This is by Shield. According to Shield, the type 1 means that type 1 dentinogenesis imperfecta means that it is dentinogenesis imperfecta which is seen in osteogenesis imperfecta, that is associated with the osteogenesis imperfecta. So according to him, the dentinogenesis imperfecta and osteogenesis imperfecta are coming together. Okay, that is Shield 1. Now Shield type 2 is, that is your dentinogenesis imperfecta not associated with the osteogenesis imperfecta, and type 3 is Brandywine type. Brandywine type amelogenesis, dentinogenesis imperfecta, that is seen in a particular population in Brandywine. Okay, that's the area, and a particular population has this type of dentinogenesis imperfecta. Okay, so this is the Shield classification. Now this is outdated. Okay, this is outdated. You can see this options only in very old textbooks. Okay, when you are working on questions which is very old, okay, you can see the type 1, type 2, type 3, but now the revised one, there is only type 1 and type 2, that is type 1 and 2, we have completely omitted this. Okay, the shield type 1 is not there. There is only type 1 and type 2 here. Type 1 is your dentinogenesis imperfecta, which is not associated to osteogenesis imperfecta, and type 2 is the Brandywine type. Okay, the Brandywine. Okay. Or we say Maryland type, that is a race. Okay, then we'll see it. So this is a type 1 and type 2 are the present revised classification. So this is the option you will see in the recent question papers. And how it is clinically presented. Clinically, what you can see is that clinically you can see the brown color of the dentin being shown through the enamel. The enamel is having no defect here, but what happens, the enamel is supported by dentin. Enamel is always supported by dentin. Enamel is very rigid material. It has no resiliency. Okay, whenever you bite on a hard substance, okay, the enamel compresses and the dentin is resilient and dentin will take up the force. So that's why enamel is protected from chipping off. But when your dentin is defective, what happens? Your enamel will chip off. Okay, why how the enamel chip off? Because the dentin is defective here. Dentin cannot hold the enamel properly, so enamel will chip off. Yeah, enamel will be lost, and the dentin is seen outside, and dentin have a brown color. So that is why we say brown opalescent teeth. Got it? So here we see brown color teeth, brown opalescent teeth, and it can affect both your dentition, or the primary or the permanent dentition will be affected. And it is mostly autosomal dominant. It is predominantly autosomal dominant inheritance, and it can affect both dentition, both primary and permanent tooth. Both dentition is affected. And what next? What you can see is that we see the types. Type 1. What about the type 1? And what about the type 2? Let's see the difference between type 1 and type 2. Now, type 1, that is the most common one, most common type you see, most common type we see. It is hereditary opalescent color. And here what you can see is that there is abnormal dentin is formed. Okay, abnormal dentin, you have this defect, and the abnormal dentin is formed. Okay, so what happens is that when the abnormal dentin is formed, it will fill up the pulpal space. So there is so much of dentin production. There's defective dentin is formed, and there is excessive defective dentin formation, and it will fill the pulpal space. Okay, pulpal space, it will fill up the pulpal space. Now what happens when it fill up the pulpal space? The pulp cavity will be reduced. It will cause a complete obstruction of your pulp chamber. Okay, the pulp chamber is obstructed. This is what you see in type 1. Now this opposite is seen in type 2. So type 2 is a particular type of dentinogenesis imperfecta, where you can see that it is seen in the races of Maryland races. That is a special race group of people, and it is seen in what we say Brandywine type. Okay, the Brandywine type dentinogenesis imperfecta in seen in the Maryland race. Okay, now what happens here? Here the dentin is not formed. Dentin is less formed. Here the dentin is less formed. So it's just the opposite. It's just the opposite. Dentin is very thin here. So what happens when the dentin is less formed or dentin is thin? What happens is that you can see a large pulp chamber. The pulp chamber is large. We have less dentin here, so it will look like a shell actually. So we have enamel, the tooth will look like this. Okay, I will draw a tooth here. Okay, I'm drawing a tooth here, and just a tooth, and you can see that this is the root canal, and the dentin which is formed will be very thin here. I have a very thin dentin. And what happens when the dentin is very thin? Your pulp chamber will be large. You can see that the pulp chamber, that is large pulp chamber, and that's why we call it shell teeth. It's empty inside. We can see that it is empty inside the shell. It is called shell teeth. And you can see that the pulp horns are very high. The pulp horns in such tooth, the pulp horn will be very high. So very careful when you prepare a cavity. Okay, it's high chance that you can expose the pulp horn because the dentin thickness is very less. So there is more chance of pulpal exposure in dentinogenesis imperfecta. Okay, if you're in type 2. Okay, so this is about dentinogenesis imperfecta, nothing much. You should know the difference between type 1 and type 2 and how it is clinically presented. Okay, so I hope you understood what is dentinogenesis imperfecta. See you in the next video with the next topic. Thank you.